Here we are in the environmental analytical facility in the Vincent building. As you can see, it's the whole floor is a lab, and it's spread between this and the floor above. The equipment in here is used mostly across the school, so within the agri-food, water and environment theme. But here I will be talking mostly about the agri-food. The students, MSc students within our courses, will be in touch with the equipment in the facilities at different time points during the MSc. So the first time that they will be using them and they will be having hands-on would be during the taught modules. They will have the opportunity with the teaching staff, but also postdocs and lab technicians, just to get a first point of contact with the equipment. After that, when they move into the group project, they will again have the opportunity to use different devices, and then at the last time during the thesis project, because they will have 3 or 4 months just for them to learn and be focused on one topic, is when they probably will understand and have an in-depth learning, understanding of specific pieces of equipment. Some of the equipment that the students would be working with, they can also find them in the agri-food industry. So if the company has an R&D department, they surely will face different equipments, Hplcs, all of them may be related with the food and safety aspects of food. So this is always very interesting, and here at Cranfield we believe that is crucial and very important, because then they would be able to take informed decisions whether they are doing research by themselves in industry, whether they want to continue their career within the academia, or whether they even go into medium management positions where they will need to talk and discuss with more technical people how to develop one product, how to develop a protocol. So it's always good that they understand from the inside how the equipment works and how to probably design an experiment. Here you can see we've got now open. This is one of the 6 school rooms that we have in this floor. So they are actually variable temperature rooms. You can change the temperature from 0 to 25, even 30 degrees. These school rooms are complementing the post-harvest facilities that we also have in building 54. So there we have 2 variable temperature rooms, the same size as these ones, but we have another one which is triple the size, just to carry out bigger experiments. So in this corridor we normally start all the agri-food trials. So when it's about post-harvest trials, we bring here the fruit or the vegetables, and then we store them at the different temperatures. We can mimic any conditions that they would be doing in industrial setting. Here we have a gas mixer. These boxes that you can see on the wall allow us to change the percentages of respiratory gases. So we can change CO2, we can also change the nitrogen, and therefore the percentage of oxygen that we are just putting into the storage boxes that we've seen that are staying in the cool room. So with this system and this equipment, we can just make 6 different experiments in all these cool rooms. So this is, for example, when we want to perform experiments that will do different control atmosphere settings. That it's also helpful just to understand the modified atmosphere packaging. So they give us the opportunity to study how we can extend shelf life or storage life while maintaining the quality of the fresh produce. Here what we have is a respiratory system. So this is a stable system. This equipment, what enable us is just to monitor real time what is the respiration rate, the metabolism of the fruit or vegetables that we have in the boxes. So we can link it with the storage boxes that we may have in the cool rooms. These tubes, they will go straight to the equipment, and then we can monitor in these graphs what is the CO2 that is being produced by the fresh produce. This is very important because we can in that way monitor how they are behaving, the fruits and vegetables throughout the storage, whether they are entering into senescence. So this will give us, and therefore industry, a very good insight about how each of the treatments that we apply perform. What we want to achieve with the different experiments, harvest trials that we carry out here, is just to assess what is the food quality after they have been treated under, for example, different control atmospheres, or when they have been treated with different packaging or different treatments to prolong shelf life. So what we do is just we take the fruit or vegetables, and then we can assess, for example, firmness. Firmness is a parameter which is very important for industry. It is very important for us as a consumer. So when I go and buy some strawberries, I want them to be firm at the time of purchasing. So a way that we have here in the lab to assess the firmness is by using a universal testing machine like this one, in this case from Instron, the company, and we can objectively and constantly assess how is the firmness of whatever product we are looking at. But then another quality attribute that we might be interested in looking at is the color. It's very important, again, how the food looks like. We don't want to go to subjective, it looks okay, it looks red, but not that red. So we've got a scientific camera with a photo box that will give us an homogeneous light conditions, and then we take images of the fruit and vegetables at each point throughout the storage period. So we can obtain parameters which are lightness, chroma or hue angle, and then by looking at them and analyzing them, we can understand how the fruit is doing, how the senescence is going, or how ripened it is, in a, again, objective way. We are now in the HPLC suite. We have moved from the second floor to the third floor. Here we've got a number of HPLCs, so high liquid performance chromatography. Once we have extracted the compounds from our food matrix, researchers but also MSc students from any of our agri-food courses will come here to analyze the chemical profile of the food matrix. So everything will end up in a vial like this with these different HPLC which have also different detectors. You can quantify compounds such as sugars, so you can identify specifically how much glucose, sucrose or fructose a matrix has. You can also look at organic acids, so very important, for example, if we want to monitor how vitamin C is developing throughout storage. And we can also look at the quantification of phenolic compounds. So we can therefore look at the antioxidant, how they again develop and how they deplete under different post-harvest treatments or any treatment that has been applied. So we can do that using these HPLCs. We have another suite where we have HPLCs coupled with mass spectrometry. In this instance, we have a higher sensitivity. We can look at compounds which are in a much lower concentrations in the food matrix, for example, hormones. Hormones as such won't give us a direct understanding of the quality, but we can understand by looking at the hormones how the metabolism goes and therefore how that would affect the shelf life or a product. We can also look at the safety of the product because LCMSs will allow us to quantify mycotoxins. Mycotoxins are secondary metabolites that are produced by specific fungi, and they can put human health at risk. Sometimes these mycotoxins come in a very low concentrations, and the LCMSs give us the opportunity to quantify such compounds. We are here in the volatile laboratory, the gas chromatography suite. We've got a number of GCs, but also GCMSs. In food, it's very important also to understand how the flavor develops. And for that, the volatiles that we can get from them they are crucial to understand their quality, but also the safety of them sometimes. For example, the typical volatiles that are characteristic for an ripened apple. Some of them are characteristic for a potato maybe that is getting rotten. So all of that, it can be studied and quantified using these equipments that we have here. We are in one of the molecular biology labs. We've got a number of PCRs and real-time quantitative PCRs. We will use these equipments to look at the molecular biology of plants and microbes. With what we do here in agri-food, we've been looking at how the safety and the quality is maintained, but we also want to understand the mechanisms behind that, which are the genes that will change and how the expression will change depending on the treatments that they have been treated with, or basically when we are looking at one specific crop which has different traits, let's say yield or resistant to a specific disease, we can use the molecular biology to understand which of those genes are highly expressed in one crop or the other. And that will be used to inform the breeders of how to breed new cultivars with specific traits of interest. We are in the Applied Microbiology and Mycology lab. As important as food quality is, of course, food safety. So in this lab, we've got some Bioscreens. The equipment that I've got by my side is a high throughput platform by which the Applied Mycology group has developed a protocol to test different growing conditions of filamentous fungi. So this equipment enables us to test a big number of culture media, antifungi, and different conditions. But if we were to do it with traditional petri dishes, it will take much more time. Together with this equipment, we also have laminar flow cabinets that will be used to manipulate fungi samples or any microbiological sample within the agri-food field.